thanks for the invitation, Cinzia, and Arnaud will start the talk.
Yeah, I mean, I would like as well to thank Cynthia for this kind invitation, and for me, the opportunity to discover your forum. That sounds pretty much attractive, and I'm really happy somehow to hold the session together with Fabia, in order to depict, I mean, some of the stories that we have been experiencing together since we are working in the field for more than about 10 years, I guess.
Yeah.
So basically, I mean, today, I mean, we will do our best to guide you through relevant industrial problems that me, as a pharmaceutical scientist dealing with solid state, has to solve on a day-to-day basis. And basically, we will be bringing you in the field of crystalline salt, crystalline form for pharmaceutical ingredient, and share some of the challenges that we may encounter. So as outlook of the presentation, we will give a really short introduction about the role of solid forms in pharmaceutical development. I will guide you through some expectation from health authorities, and we will be introducing some relevant elements in terms of limit of detection and limit of quantification. And hopefully, I mean, we will trigger your interest through two different case studies where basically, I mean, we had to identify and to quantify some crystalline impurities, which had some impact on drug product performance. And also another very interesting case of salt disproportionation, where the active pharmaceutical ingredient is reacting with some of the excipients. And for concluding, we will be sharing some perspective on ongoing activities and eventually what will be happening next. During this talk, I mean, I will be talking about drug substance. I mean, by drug substance, what I mean is the active pharmaceutical ingredients, so the chemical structure which has the biological activities, and I will be talking about the drug product. I mean, the drug product is the drug substance which is presented into its pharmaceutical forms and that the patient are experiencing. It can be tablets, it can be hard gelatin capsule, but also the type of drug product, depending on the type of administration and the disease area. So as you may know, and as you may expect, I mean, in terms of new chemical entities, so the landscape is certainly changing a lot over the last 20, 30 years. I mean, and this is mostly due to the fact that the molecules are getting more and more complex. I mean, all the easy space has been eventually discovered. And despite we are discovering some new molecular targets, which may eventually repurpose some of the old drugs, so the physical properties of these more complex active pharmaceutical moieties is getting more and more difficult. And this is translating into change in terms of physical properties compared to what we were used to develop in the past. So most of the drugs are getting poorly soluble, and quite often they do have as well some low permeability, meaning that even if you bring them in solution, basically, I mean, they are encountering some challenges in order to move from the gastrointestinal tract into the systemic system. In order to help the different pharmaceutical scientists, but also the health authorities, in order to categorize the drug, so a biopharmaceutical classification system has been established. And this aims somehow to classify the different drug substance in terms of permeability and solubility. And as you can see, I mean, from this graph, I mean, most of the drugs are getting practically insoluble. In order to help with the dissolution of those drugs, I mean, we need to develop some special delivery system. So the idea is to trick the physical properties of the drug substance in order to make them soluble into the GI tract, the gastrointestinal tract. But certainly, it requires to fix the nature of the solid form. I mean, talking about the crystalline form itself, but also eventually the salt form as earliest as possible during pharmaceutical development. More and more importantly, so patient centricity, I mean, is getting more and more relevant. I mean, by means of patient centricity, we understand the convenience for the patient to take the drug. So one may minimize the pill burden. Unfortunately, everybody of us has been experiencing some large pill, and we try to minimize the size of the pill as best as we can. And we have certainly also to fulfill some regularity requirement. So the health authorities, I mean, have been establishing some ICH guidelines, which tends to summarize the expectation across the whole different health organization across the world. And certainly, if you are talking about relevance of the solid forms, the chapter Q6A has some relevance and importance. And certainly, it is expected that we are working more and more in terms of quality by design. We are using process analytical technologies, and there is certainly some need to have a robust and proper control of the manufacturing of the drug substance and drug product in order to ensure the desired quality of the final product and the safety ultimately of the patient. Talking about the different solid forms, I mean, it's really obvious if you change the different polymorph that you may change the different properties. I mean, this is what I tried to exemplify here on this nice slide, dealing for different polymorphic forms. So they may have different crystal habit, but may have also different color of powders. I mean, there are not too many examples with such a nice change of colors, but it does exist. But certainly, if we think about a crystalline and polymorphism landscape, one may think also about circumventing, I mean, those type of changes by forming salt when the drug is exhibiting some acidic or basic moieties. And as you can see, similarly to a change in crystalline form, I mean, we have significant differences in terms of properties on this slide. I mean, I'm talking about crystal habit. Again, in terms of pharmaceutical development, I mean, the chemists are working on optimizing the chemical scaffold of the active pharmaceutical ingredient in order to make sure that it has the highest selectivity, the highest affinity with the target, but also some optimum physical properties. And quite often, I mean, they are reaching some limits in this field. And this is to us, I mean, as a solid state scientist, I mean, to work around this chemical compound, I mean, depending if they do have some acidity or basicity function, we may consider to characterize a free form, but also we can try to form some salt. And if none of those properties are meeting our requirement, we may think about looking on co-crystals. By means of co-crystals, I mean, we understand by bringing this chemical compound together with a conformer and build some crystalline forms where basically two components are interacting together with non-ionic interactions. Certainly, I mean, in all these different categories, we can deal either with crystalline form or with amorphous material, and they may exhibit or not polymorphism, but also pseudo-polymorphism. And this is the type of landscape that we have to investigate and where we need to take a decision on which form, in terms of chemical composition, the free form, the salt, or the co-crystals, either as crystalline or amorphous material, but also to consider the target crystalline form in terms of polymorphism or pseudo-polymorphism. And this turns to be super important because all of these individual forms, they would have different physical properties. And maybe if you are going on the next slide, you will get an idea about the different physical properties that can be impacted. I mean, certainly the solubility is of primary relevance there, but also the dissolution rate, the dissolution kinetics, since they will be affecting the bioavailability, but also the toxicity. If you have a huge peak of concentration after administration, so the crystals may change color, they may change habit. This will be certainly impacting the processability of the drug substance in terms of filterability and drying, but also the drug product manufacturing. They will have also different melting point, different hygroscopicity. I mean, how much is sensitive the drug, depending on the exposure to relative humidity. And certainly, ultimately, a very important parameter is the chemical stability and the compatibility with the excipients. Ultimately, I mean, we do want to build some drug product, to engineer some drug products which are exhibiting the key quality attributes that we want to design in order to meet the target dose, the target dosage form, and as I was mentioning before, to minimize the patient burden in order to have a drug that can be easily administered to the patient, or let's say as easiest as possible. And very importantly, I mean, we need to do this in a way that we are able to control and to make sure that the drug product at the end, I mean, is showing the adequate performance, but also the adequate safety and efficacy. By means of safety, we mostly understand what is the concentration into the systemic system, but also we need to make sure that upon aging, I mean, we don't create some chemical impurities that may have also some biological activities. As I've been mentioning before, I mean, all these things have been describing in the ICH guidelines. And if you are talking about solid forms, I mean, we are mostly focusing on Q6A. And the expectation from health authorities are clearly depicted. So we need to conduct some polymorphism screening and to come to a conclusion if different crystalline forms does exist or not. If yes, we need to characterize them in terms of physical properties, but also in terms of solubility. And we need to understand, I mean, what are the impact of the different forms in terms of safety, efficacy, and performance. In case we understand that those would have some relevant impact, I mean, we need to set up some acceptance criterion on the drug substance level, which means some analytical methods which are running under quality framework in order to make sure that we have a good control about the quality of the drug substance that we are manufacturing. If you think about what is the impact on the drug product, I mean, again, we need to understand if the drug product would be sensitive or not to the change in drug dissolution performance. If yes, we need to establish some criterion. And if the drug product dissolution test is not sensitive enough, one may have to think about setting up some acceptance criteria, eventually with more direct method, eventually X-ray powder diffraction, in order to have a control on the drug substance level during the drug product manufacturing, but also during the release and the stability and ultimately the shelf life of the drug product. You can imagine that doing all these things, I mean, talking about solid state might turn to be a bit challenging. And we, as originator, basically, we tend to favor the development of the most thermodynamic stable form. So basically, in order to gain a certain level of confidence that absence of solid form will be happening during drug substance and drug product manufacturing. To give you an idea about how our labs are looking like, so you have here on this slide a collection of methods that we are using on a regular basis. I mean, talking about solid state, I mean, one may certainly name X-ray powder diffraction, but we are also looking on the hygroscopicity, thermal behavior, thermogravimetry, where basically we are looking on the loss weight upon eating, but also DSC, different scanning calorimetry, in order to understand potential changes upon eating. We are applying some spectroscopic techniques, mainly infrared, mid-infrared and Raman spectroscopy in bulk or in microscopy modes. So when we have some light microscopy on the top of our hand, together with scanning electron microscopy. But we do also use quantum mechanics and molecular modeling in order to understand what will be happening to our drug substance. Certainly, we are able to characterize them by solubility and dissolution kinetics. And in case we are interested into crystalline content, we may use microcalorimetry. Very importantly is that some of those techniques, the ones that have been highlighted in green, can be operated under GMP, these good manufacturing practices. I mean, this is the general quality framework that the pharmaceutical industry is using in order to ensure that each of the methods is showing the right level of robustness, precision, accuracy, and can be transferred from one side to the others in order to make sure that all the eligible labs can conduct the same test and come up to the same result. As you can imagine, so we have certainly a huge variety of analytical techniques, but from time to time we need to extend our toolbox to more sophisticated techniques, and I've been listing a couple of them. It can be synchrotron powder diffraction, but also synchrotron single crystal diffraction, small angle X-ray scattering. Electron diffraction is retaining a lot of our attention at time being, but it can be as well solicited NMR spectroscopy and many others. Also, I believe that I've been setting up the scene in order to introduce you to our first case study, which is an investigation on a controlled release formulation. So talking about controlled release formulation, the product has been designed in order to address this patient centricity. So meaning that instead of asking the patient to take the medication a couple of times in a day, we were developing a type of pill which is releasing continuously, the drug substance over time after its administration. We were certainly running the expected polymorphism program, and basically we were understanding that the drug was forming, was exhibiting polymorphism. And in this case, we were selecting the most thermodynamic stable form on the drug substance level in order to minimize the burden of quantitation exercise as a drug product. In terms of product design, we wanted to get dissolution control release over a period of time of 8 hours. And just to give you an idea about what this test is, it is described in the monograph USP2. And basically, it is a large vessel with a volume of about 900 milliliter, which is temperated at 37 degrees. It is equipped with a paddle. We are introducing the drug product in the sinker, and basically we are monitoring the increase of concentration over time during a period of time of 8 hours. And for this product, we are designing as a specification attribute that we wanted to have a release concentration between 20 and 40% after 60 minutes. Our team of formulation was designing the drug product, mixing it and engineering the different excipients in order to achieve that target. And you get an experimental dissolution profile with the curves with dashed point in yellow. So on the x-axis, you have the time profile. On the y-axis, you have the percentage of drug which is released into the media. And everything went well during the clinical trials until we were identifying that some of the product and some of the batches was showing unaltered properties when the drug product was exposed to thermal stress. And this is depicted by the dissolution profiles that we are collecting with a black trace. one may ask ourselves, I mean, why 60 degree? I mean, it is eventually some conditions that are unlikely to happen during the drug product storage condition, but eventually if you want to coat the material, to coat these tablets, I mean, with a film coating, we need to operate at elevated temperature, and this is what we were looking at. And as you can see, after 60 minutes, I mean, the profile, I mean, was eventually close to fail the specification. We were then starting to do some investigation. We are calling them root cause analysis. And we were sitting all together with the different scientists of the team, trying to understand what is happening. We were looking certainly on all the different key quality attributes, and none of the others was affected. And then we were starting to do our investigation. We are looking using X-ray powder diffraction, mid-infrared spectroscopy, and all of the techniques that I've been listed before. But expect this change in terms of dissolution performance profile. We were not able to identify any defect or change compared to the unstressed sample. And this remained true until we were running scanning electron microscopy. I mean, scanning electron microscopy on drug product level, I mean, is a bit unconventional because we need somehow to break the tablets and get it under the scanning electron microscope, put it under vacuum. But then it turns really obvious that something was happening. So as you can see, the initial material, I mean, is only showing some smooth surfaces. And on the different samples that we are identifying, we could see the appearance of needle-like particles. As a solid-state scientist, I mean, needle-like particles, something should be happening in terms of crystalline forms. We were looking back on our X-ray powder diffraction data. We are trying to increase the data acquisition time in order to find out what the problem was, and this without any success. As a matter of fact, we were wondering if we are just blinded because of the limit of detection of our analytical instrument. Putting all the information together, I mean, dissolution kinetics was affected. No other obvious attributes were somehow affected, except the appearance of these needle-like particles by means of scanning electron microscopy. And we are looking on the properties of the drug substance itself. I mean, it shows polymorphism. We were selecting the most stable form, and we knew that one hydrate could exist, and that this hydrate has much lower solubility compared to the form that was used. And also the morphology of the hydrate, I mean, was known to show needle-like particle. So we are telling ourselves, okay, so we understand what the problem is. Eventually, we don't understand why the hydrate is formed. But before going into the preventive actions, we need somehow to demonstrate by direct means that the drug product is getting contaminated with the hydrated form. And this is where basically, I mean, I was taking my phone, I was giving a call to Fabia and asking her if something can be done on the drug product level.
So, so I think I take over here. So I will eventually tell you the rest of the story, but maybe before doing that, let me just step back a little bit and tell you some about the technique that eventually we applied to solve Arnaud's problem. So the technique is going to be a synchrotron X-ray powder diffraction. And so since we understand that the audience is quite broad, we decided to give you a little bit of an introduction to the technique. So the principle of the X-ray powder diffraction is here schematically depicted. So you essentially send a source of X-rays, monochromatic X-rays, onto a sample, which in our case is either a powder sample or even an intact tablet. And then you collect the X-rays that are scattered out of the sample, and you generate X-ray diffraction patterns that are a little bit as the one here as given as an example. So you collect the scattered intensity as a function of the angle of scattering. Now, traditionally is a very traditional technique, and Arnaud mentioned that is quite used in the pharmaceutical companies in-house. So you can use an X-ray tube as a source of X-rays, or you can use a synchrotron facility, like as a giant source of X-rays. The technique is the same essentially, but the exceptional properties of the synchrotron facility enhance the property of the technique itself, and the point of this discussion will really be that. Now, for those of you who might not be familiar with synchrotron radiation, then I will just briefly tell you what it's about. So a synchrotron radiation is electromagnetic radiation produced in synchrotron facilities. These are particle accelerators that consist of a linear accelerator where electrons are thermally generated and accelerated into a sort of a pre-storage ring, a booster, where the energy, the final energy of the electron beam is achieved. And then when this happens, the electron beam is injected in the storage ring, and the orbit is finally optimized by a very complex array of magnets. Now, there is a classical principle of physics that says that charged particles that are accelerated emit electromagnetic radiation. And this is what happens. Now, when the velocity of the moving particles, in this case the electrons, is much smaller than the speed of light, then the properties, this emission is quite isotropic around the moving charged particle. However, when the velocity of the moving charged particle reaches values that are close to the speed of light, then the properties of this electromagnetic radiation change dramatically, and it becomes very directional. If the parameter that governs this directionality is the gamma parameter, that is the electron energy in the electron energy, rest energy of the electron. So here, just to give you an example, this value is approximately 2000 times the energy of the electron in GeV. So is a number, of course. And then, for example, for the Swiss light source, which is a medium energy ring, 2.5 GeV, this is approximately a factor of 5,000. And so this gives you an idea of how this directionality changes. Now, in case you are interested in listening more, then I saw that there is still available a video that is available on this site in case you are interested in. And I gave a lecture a long time ago on this. Now, so back to this slide. So this how these elements look in real in the example of the Swiss light source. So the powder diffraction station at the Swiss light source, where I've been myself responsible for many years. And then, of course, a detector that is used to collect that scattered intensity. Now, what I very much like to summarize is that in order to to do synchrotron X-ray powder diffraction efficiently, you essentially need three ingredients. You need an efficient synchrotron facility and beamline optics. Then you need a state-of-the-art diffractometer and outstanding detection systems. So among the properties of synchrotron radiation, those that are particularly pertinent to synchrotron XRPD are the extremely high intensity of the beam that allows us to collect data very rapidly, and also to afford detection systems that are consuming photons very much. Then tunable photon energy is also very convenient because we can make the choice of the energy to be used so that we can optimize the background. And then also is linearly polarized in the plane of the orbit, and you might understand why the diffractometers in a synchrotron facility are placed vertically, is because then you don't need to make any correction to polarization, because you have no dependence of the angle as a function of the polarization. And then the state-of-the-art diffractometers is very important to have a great mechanical stability so that you can achieve extremely aggressive resolution and accuracy and precisions of the order of few arc seconds. And then also it is quite important as synchrotron to get a high flexibility to accommodate all kind of sample environments, and this has been certainly the choice of the Swiss lessors. And then for the detection systems, there are many appropriate to the purpose. The one that we use massively is our solid-state silicon detectors, a mutant detector. So for the data that I will be discussing today, they are extremely fast and efficient, and they are particularly optimum for the study of pharmaceutical compounds that suffer from radiation damage. So acquisition are extremely efficient. We can cover a wide angular range in a shot, so measurements are very fast, and also are all angles are recorded at the same time. They are characterized by a very high inherent angular resolution that, of course, needs to be convoluted with the size of the sample, because these are displaced detectors, but still is quite impressive. And they have a very high dynamic range, which means that you can detect at the same time extremely elevated signal and very small signal, which for trace analysis, which will be really the subject today, is very important. And then single photon counting means that you can set up a threshold, and so you can really clean the data from a background. Okay, so back to this slide. What are the information that we can extract out of a X-ray powder diffraction pattern? Well, from the position of the diffracted peaks, we get information about size and dimension of the unit cell. From the intensity ratios, then we can get information about type and location of atoms in the unit cell. And by looking at the full width at our maximum of the diffracted peaks, of course, at the condition that your instrument does not contribute to that width, which makes angular resolution very important, then you can get access to the intrinsic properties of the material, so-called microstructural analysis. So this is essentially what you can get out of such diffraction patterns. You can study the structural and microstructural properties of materials. Now, Arnaud has very well discussed the relation that exists between the structural properties, so the crystal form that you are dealing with, and the physico-chemical properties of materials. So this is what is called polymorphism of substances, which is a very important topic in pharmaceutical science, which is the ability of substances with identical chemical composition. In the case of pharmaceutical, the identical molecular structure should crystallize according to different arrangement of the atoms. And Arnaud explained us how those forms can influence the physico-chemical properties of materials. So you here see an example of bupivacaine hydrochloride, one of the first molecules I've studied in the past. So you see the same molecular formula can crystallize into very different crystal forms. And what is important, as we said, is that the structure influences the pharmacological properties, so how a drug will eventually behave. And this is quite important, therefore, to have a technique available that is capable of distinguishing very well and directly. So allows us the direct, and I call it the unquestionable identification of different polymorphic forms. So here is the diffraction patterns of these two forms. So you can see that there are many peaks that are in common, but there are also peaks that uniquely identify a form, what I like to call testimonial peaks. So that's the reason why at Excelsus we very much concentrated on pushing this ability of detecting very small traces of a pharmaceutical crystal form, and also we spend a lot of energies in trying to refine the methodology to quantify very small traces. So this has been the first attempt back in 2013 to see what was the ability of this kind of detection system to detect small traces. And so you see that here in a mixture of active pharmaceutical ingredients, we were able to go down to better than 0.05% in weight, which I think at that time was the best circulating. And then later, by tuning the optics differently, we were able to reach better results. So essentially, we decided to test whether the level of detection could be enhanced by focusing the optics very much, so underfilling the sample and focusing the optics onto the detector. And that was a good idea. So here, if I remove the reference here, you see that in unfocused or classical optics conditions, we were able to detect only one of these peaks at those very small percentages, but more than one in these. And then here in this other example, there was even more striking, because in unfocused conditions, we could not reach anything below 0.1%, but in focused conditions, we were able to detect several peaks at 0.01% in weight. So it means 10 times better. Recently, we also challenged ourselves in dealing with protein powders with a collaboration with the group of Irene Margiolaki at the University of Patras. They are really pioneer in the application of Synchrotron XRPD to protein powders. And we apply these methodologies we developed at Excelsus to measure their proteins, so far measured at ultra high resolution with multicrystal analyzer detector. And for the first time, we were able to detect a second phase of human insulin here. And then they also made a comparison between their lab data, the ESRF at the multicrystal analyzer detector, and our data at the SLS. And then you can see that although the multicrystal analyzer detector, you can access higher angular resolution, that the difference is not striking, and we can reach extremely higher signal to noise. And now finally, I come to where Arnaud left the story. So as Arnaud said, he contacted us, asking whether we could have elucidated the origin of these needle-like. And specifically, as Arnaud already said, they were suspecting the presence of a hydrated form of this molecule, and they were able to give us the reference for that hydrated form. So we collected data, Synchrotron XRPD data, and we saw peaks that were not, we couldn't detect any of the hydrated expected form, But we detected other peaks, and now here appear quite evident, but these are really zoomed in a lot, so are very, very small peaks, and even in some samples even much smaller. And these peaks were not explained by this hydrated form. We had access to those SEM nice pictures, which were showing that morphology needle-like. So back to the literature, then we could figure out that there was another form. There was an anhydrous form that also had that morphology. And again, here the power of a tool that allows you a direct detection of form. These peaks that we were detecting were really matching perfectly the structure of this other form. And I think Arnaud, with that information, could continue the understanding.
And in fact, the team of Excelsius was not only looking on identifying what was the form that was contaminating the sample, but they were also able to develop a semi-quantitative model in order to evaluate the ratio between the alpha and the beta forms that we were intendedly putting into the formulation. This has been remarkable because up to now, I mean, the way how you are able to do a quantification into a physical mixture of forms, I mean, is either to do some Rietveld measurements or to do some spiking experiment in order to understand what is the extent of the content of the other forms. But here, basically, I mean, one has to keep in mind, and I apologize, I was forgetting to mentioning it, that we are dealing with a drug product which is containing plenty of other excipients. So, I mean, Synchrotron, I mean, was extremely helpful on one end to identify, let's say, the nature of the additional peaks, but also to make sure that we are getting the right specificity against the other form of the drug substance, but also including all the additional diffraction elements of the crystalline excipients that were used. And basically, I mean, Fabia and team, they were able to evaluate what is the ratio between the different forms. And we were realizing that there was a quite nice correlation with the impact on the dissolution performance. And as you could see, I mean, we were sending a huge variety of samples. I mean, starting from what we believe to be pure sample up to some samples that were strongly affected by the disease that was propagating into those different batches. And they are nicely correlating with the loss of dissolution performance together with the evaluated content between alpha and beta form. Important also to note, if you would have been relying on the SEM picture, I mean, certainly we see that something is happening, but it would have been really difficult to evaluate that eventually the sample, which is highlighted in orange, is containing about twice more amount of material of this contamination compared to the sample in red. So here, I mean, use of synchrotron X-ray diffraction was extremely powerful, not only to identify what was the nature of the contaminant at the end that was impacting the dissolution performance, but also to assess almost quantitatively, I mean, what was the extent of the propagation into the drug product.
Yeah, and I'm glad, Arnaud, that you underlined the fact that these were drug products. So it means that besides these crystal forms, there was a mess of peaks. And so we indeed developed a way to model all the rest, which was not contributing to those competing forms, to stabilize the quantitative analysis, indeed.
And somehow it was not the end of the story, because we were expecting somehow that Excelsius would be highlighting the presence of the hydrate, I mean, which was known to have a significant impact on the dissolution performance. But on the other hand, I mean, what we have been highlighting there is that the product was contaminated with a metastable form. And it was creating, at least to me, I mean, a lot of headaches, because in principle, a metastable form, I mean, has higher solubility compared to the most stable form. But we were contemplating a loss of dissolution performance. And somehow, I mean, all this investigation were leading to the understanding on why this alpha form, I mean, was getting was developing into our sample. I mean, again, there is no opportunity to understand that a stable form would be converting into a metastable form, but it was conducting us to understand that during drug product manufacturing, depending on some of the process parameters, we were producing a small amount of amorphous material, or small amount, an amount of amorphous material that was serving as a kind of reservoir of the compound to crystallize into the metastable form. So basically and ultimately, I mean, at the end of the story, we were able to understand, I mean, what was impacting the dissolution performance on one end, but also we are able to go back into the drug product manufacturing design in order to understand, I mean, what was creating some lack of robustness during the DP or drug product manufacturing. So now we will be guiding you to a second study case, which is nicely exemplifying as well another application of synchrotron powder diffraction, which is the role of trace analysis in the context of API, active pharmaceutical ingredient, and excipient reaction. So as I was mentioning before, I mean, we have the active pharmaceutical moieties. Here I was just taking the example of imatinib, which is a drug that showed a very good efficacy in some of the cancer patients. The API, in order to get the right physical properties, was used as a mesylate salt. And then this white powder, I mean, has to be converted into engineered tablet to show, I mean, the appropriate performance that we are expecting. And certainly, when you are doing this engineering of the formulation, I mean, you want to enable your drug in order to meet the expected performance of the product. But something that you certainly don't want is that the drug does react with the excipient in order to create some chemical impurities. And typically this goes via an exercise which is so-called formulation design. There are many general principles that are, let's say, establishing the foundation of the screening approach. And one of the really first things that the teams are doing is to look on the chemical compatibility between the drug and the excipient. In order to be smart and somehow to anticipate what could be happening at later stage during the shelf life of the product, we are using accelerated predicted stability programs, where basically we are increasing stepwise the temperature and looking on the chemical degradation of the product, but we can also add the relative humidity as a kind of additional stress condition. Typically, I mean, most of these experiments are exclusively focusing on chemical degradation, because it is well understood that chemical reaction can get fit based on Arrhenius principle. But nevertheless, there is certainly a need to look on physical incompatibility, or let's say to identify physical incompatibility between the drug and the excipient. And one of the challenges that we are facing again as a solid-state chemist is that those type of reactions are not following the same kinetic rules. I mean, it's not about mixing A and B. And it's not only about activation energy and constant of reaction rate, but basically we need to understand, what would be the impact of reactive spots that can be related to high surface energy, depending on the way the drug substance has been manufacturing. But as we have seen before, what is the extent of the amorphous fraction into the drug product that may lead to such a kind of physical interaction. In addition, if you think about X-ray powder diffraction, we are looking, let's say, on the generation of a crystalline form, and certainly the byproduct may have also to undergo via nucleation kinetics. In this case study, basically we have been focusing on HCl salt, an hydrochloride salt of an active pharmaceutical ingredient, and we were a bit suspicious about the selection of this salt. The salt was showing really good biopharmaceutical performance, but we knew that the API has a quite low basicity. So meaning that, yes, fair enough, we are able to produce some hydrochloride salt, but on the other hand, we were understanding that the strength of these ionic bonds, was relatively weak. And then we were certainly looking with a lot of curiosity and trying to understand as best as we can, what was the potential interaction with the excipients. We were certainly looking also on chemical stability by means of chromatographic techniques, but physical stability-wise, we are looking on our lab XRPD instrument by increasing the data acquisition time and asking ourselves if the free form of the API could be detected. And in this case, we are not able to see any formation of the free base, but being very suspicious about the low acidity of the compound that was not showing any obvious weaknesses, even when exposed to high relative humidity, we were asking Excelsius to give a look with a different magnification lens on those samples.
Yeah, and again, we received there was available a reference for this free form, so we performed the measurements at the highest trace analysis, trace detection conditions, but we couldn't see any of that free form indeed. However, looking at the data, there was something going on. And again, the power of a technique that allows you a direct access of the crystal forms that are present in your sample is extremely powerful. Because what we saw that something was changing. It wasn't the free base. Look at here, the free base reference. There was absolutely nothing in there. But we saw that something was going on. And so the idea we had was to try to interpret that trend, that observation, try to see whether there was a trend, essentially. And so not knowing what was going on, then we decided to perform a normalization of the intensity to a reference that we assumed being the crystal form, the desired crystal form, which is this API, these green peaks. And then we saw that indeed there was a trend. So a signal that was like decreasing, and the signal at the same time was increasing. We understood from Arnaud what was the sample, the signal that was decreasing. It was sodium stearate fumarate. And so there was a sort of a correlation between a consumption of the sodium stearate fumarate and something that was growing. Furthermore, we also noticed, and here the power of having data with a very low noise, then there was some signal above expected at high angle. And then for pharmaceutical compounds, you have a structure factor that is over in that range of 2 theta values. And so it was quite surprising to see peaks at such high angles, and we could identify them as being sodium chloride. So of course the reaction was immediately to ask, to describe what was going on to Arnaud. And maybe Arnaud, I just let you continue the story. So essentially, Arnaud made these model. So he immediately understood that this could have been a sign of interaction between the salt and the sodium stearyl fumarate, correct?
Yeah, so I guess, as you said here, the very important element was the formation of sodium chloride. So then we were looking, with the formulators, on the composition of the material, and eventually considering as well potential impurities in the different excipient. But it was turning really obvious that the only source of sodium, was coming from sodium stearyl fumarate, which is used in a very small amount as a lubricant of powder in order to improve the flow properties. And then, speculating that sodium stearyl fumarate, is entering into the game and is somehow contributing to bring into the chemical reaction, the sodium element. It made a simple tilt that the source of the chlorine, must have been the drug substance. And that, okay, eventually, the freebase was not detected, but using the same methodologies than before, it's not because we don't detect it that it is not present.
Yeah. But then, of course, we needed to understand, so if I go back, here, then of course, in order to prove Arnaud's model of what could have been going on, then we needed to demonstrate that we detected stearyl fumaric acid. And I remember that there was something growing up while sodium stearyl fumarate was going down. And so we needed to prove that these peaks, additional peaks that we were observing, were indeed stearyl fumaric acid. And so at that point, there was no available reference in the literature, even for purchasing. And so I'm extremely grateful to one of Excelsus collaborators at that time, actually the first collaborator, Paolo Mazzeo, who was a very good chemist, and he had the idea to generate some of these stearyl fumaric acid by liquid assisted grinding. And so he did that in the middle of the night. And then we measured that right away. Oops, sorry. And then the signal was absolutely matching perfectly. And so that was the end of the story. So proving that that model was really explaining the data. And so as Arnaud already said, the fact that we were not able to detect free base wasn't really a proof that there was no problem going on, because it could have been below our level of detection. Consider that 50% of the free forms in an amorphous or poorly crystalline form, and so this of course compromises the detection. But this wasn't a reason not to have something going on.
Yeah, and I guess from the customer experience, what was really powerful there is that we are coming with a problem statement, and instead of only focusing on what we are expecting to see, to get this scientific discussion about the observations that are made, and use somehow this surrogate product in order to understand what is eventually happening on the drug substance, which is turning to be relevant in terms of health authorities' expectation, but also to understand what can be the potential impact. If this reaction isn't propagating to a larger extent, and if it is affecting the other quality attributes that have been designed into the product. So and here, I mean, using some of this sodium chloride, I mean, which is eventually a really good scatterer, I mean, allowed us to anticipate some unwanted problems at the right time in development.
So I think that now we are almost arriving to the end. We were thinking just to give you an idea of what else, what other developments are ongoing, many of them involving either Novartis or other industrial customers. So I was mentioning the importance of accurate quantification. In the past couple of years, we have been developing further methodologies to enhance the accuracy of the quantification. I mean, it is not always possible to perform quantification based on written refinements, especially if we are quantifying very small traces. It is not always possible to have a very good model. Not only, even if you are able to apply these classical methodologies, then you have to make sure that the subsampling of your sample is really representative of the physical mixture that you are attempting to quantify. And for pharmaceutical compounds, it happens very often that there is a volumetric inhomogeneity of the distribution of undesired forms that compromise the quantitative analysis. And so we developed a methodology that allows us to completely be independent of the homogeneity of the distribution of the undesired forms that we call the control net mass method. I'm not going to go into many details, but it's essentially a methodology that relies on the accurate weight of very small quantities of materials and the measurements of the entire weighted masses. And so quantification is really relying on the precise measurement. And so here I just show you just one set of patterns where we were quantifying in this way. And then you can see the trend was quite impressive. And this is a poorly crystalline material. Then the other developments are the combination of SAX and SXRPD, and this involves also Cinzia Giannini and group, as well as Novartis. And indeed, we recently published a model developed by Liberato De Caro that allows us to model the size and shape of drug delivery system that were of interest of Novartis. And it was a quite interesting work that we are now continuing into a longer term project. Then we are also interested in the combination of electron diffraction and SXRPD. So with Arnaud and Nanomegas and Eldico Scientific, we are trying to improve and put that in a tool, in a powerful tool. We are also collaborating with Rocco Caliandro at the CNR in Cinzia's group for the quantification of amorphous mixture in dual space. Then high throughput applied to pharmaceuticals is something that we are testing with Nicola Casati at the Swiss Lab Source. Per distribution function for the characterization of poorly crystalline materials is a multi collaboration with multiple groups. Analysis under pressure. Then we work a lot with, for the intellectual properties, so legal cases. and then also advanced ab initio structure solution, which is the field of expertise of Pamela Whitfield at Excelsus. Then there is, I mean, all these are going to, all these collaboration involving Novartis are probably going to increase. And maybe, Arnaud, you want to comment on these?
Yeah, so I mean, I guess, I mean, one of the elements that we wanted to share with you is the importance of the scientific discussion. I mean, even in a kind of service-based interaction, and out of let's say unexpected type of services, I mean, there is some great science happening there. And this great science is basically capitalizing on the interactions that we have as scientists. And since 1st of November last year, I mean, the Saint-Ouen Basel campus, where basically, I mean, Novartis has its headquarters, I mean, has been opening its door to startup and to company fostering innovation. And this went together with the building of this really nice pavilion. So I don't know if you are looking on it. I mean, this is this nice ring, which is close to the Rhine. And as you have been nicely understood, that I'm an early adopter for application of synchrotron radiation for pharmaceutical powders. And some of my colleagues were asking me, I mean, if we are building basically our own synchrotron facilities. I mean, it's certainly not the case. I mean, it's only the visitor center, which is really nice. But nevertheless, I mean, it was triggering the idea to foster and somehow to see if there is any opportunities that Excelsus, I mean, may find a nice spot on the campus in order to have them close to us for fostering those type of interaction. And basically, we had a couple of discussions with Fabia with this regard. Yeah.
Yeah, and then actually, didn't take too long to make that decision to have a second location. Here, I just show you, we are just moving. We just moved 1st of March. So we have a second location. Of course, we do not leave our premises at the Parque Innovare, the Innovation Park, near the Synchrotron facility, but we have a second location that allows us to a faster and easier discussion with the pharmaceutical scientists, not only at Novartis, but in the Basel area. And I think we are just ready for the acknowledgement. So I really would like to acknowledge the work of my collaborators, in particular Mathilde Reinle-Schmidt, Michael Morin, Pamela Whitfield, Fanny Costa, and of course, former collaborators. I saw Mauro Povia participating. So Mauro has been one of our collaborators and helped us a lot with this methodology of the control net mass that I was mentioning before. Well, of course, Paul-Scherrer Institute, Swiss Light Source, Material Science Beamline, and the Park Innovare, so the Innovation Park, where Excelsus has premises. And then the institutions with which we have collaboration, so the CNR IC Bari, University of Patras, Alba Synchrotron Facility, DAISY, APS, and Purdue University, and then of course all the industrial partners and all the challenging cases. Oops, sorry, that was it.
And from Novartis' perspective, I mean, I would like to thank as well some collaborators in field of solid state chemistry. I mean, Michael Moods and Evgenia Rozaki, but also, I mean, the early adopters. I mean, because I was convinced that some of our challenges, I mean, might be solved using synchrotron powder diffraction. But I had somehow to convince, I mean, the colleagues from the team, I mean, to provide us with the funding for this. And I would like to thank more particularly Mei-Ling and Thomas Rigassi, who have been, let's say, early adopter of this kind of strategy back to to 10 years ago.
And then, of course, all of you for your attention. And I think we are done here.
Thanks a lot, both of you. And so we have, let's say, 10 minutes more for questions from the audience. Any curiosity? That's a proper time to ask Fabien and Arnaud. So with this, there's a silence now. That's typical. So just to break the ice, I would like to ask both of you. I'm sorry. So there is somebody showing up. Please.
Thank you. Thank you very much for these two voices talk. Very interesting. I would like to ask you a curiosity about an example that was plotted during the explanation of the technique. You talked about non-focused and focused aspirin with salicylic acid patterns. Could you speak a little bit in detail? What do you mean focused or non-focused scans? Thank you.
Yeah, so I was really referring to the optics, to the beamline optics. So generally, you tend not to, to use a parallel beam. But then what we, first of all, you tend to not to underfill the capillary. So we use, by the way, I didn't go into too many details, but we use between 0.5 and 0.8 millimeters capillary, and then generally you go on with a almost parallel beam, and then you do not underfill the capillary. It means you have a size of the beam that matches the capillary, and that way you always have the same projection of the capillary. So you always have the same dimension of the capillary seen by the beam. Now, the idea that we had was to focus the beam onto the detector. So this implies underfilling the capillary, but implies that you can go very close to the intrinsic angular resolution of the detector, that instead is generally convoluted with the dimension of the capillary itself. So the disadvantage of that is that by underfilling the capillary, then you have a virtual smaller size of the capillary, only at very low angles. And then as soon as you reach the 90 degrees, then you see the entire projection of the capillary. But for these kind of materials, and in particular for indexation, then this is a huge advantage, because essentially you get very, very sharp peaks at low angle. This made a huge difference.
Thank you.
Great. So if there are other people who want to ask questions, please open your camera. Otherwise, I will meanwhile ask both of you. As you know, I have a particular feeling for nanomaterials since many years. So I would like a short comment from Arnaud concerning this future of nano formulation or nano drugs, and at the same time a comment from Fabien, how difficult is to rescale your level of detection that you have shown in the case of nanomaterials when you have, let's say, nano as small as, let's say, 10 or 15 nanometer, something like that, or even smaller.
So maybe I start, with a perspective. as hopefully, I mean, you have been understanding, I mean, from the really first slide of the presentation that there is a need in order to accommodate the challenging physical properties of the compounds that we are dealing with. And in order to circumvent, I mean, the poor solubility, eventually, I mean, dealing with nanocrystalline, I mean, is one of the alternatives. And it turns to be super relevant in case the absorption is only limited by the solubility, and if it has a good permeability, and using nanocrystalline suspension, which are exhibiting a really huge surface specific area. So we are able somehow to push somehow the absorption thanks to the high dissolution kinetics and provide the right exposure. I mean, there is, since about 10 years, an increasing number of drugs on the market which are exhibiting such a kind of technologies. And certainly, I mean, back to the analytical scientist that I am, I mean, it asks a couple of challenges. I mean, how can we control the solid form purity of a nanocrystalline material? What is happening to it when we are isolating it and transforming it into a drug product? And certainly, I mean, there is an increasing need of tools and technology in order to be able to properly address those questions.
Now, regarding your question on detectability of poorly crystalline materials, of course, the level of detection is not an absolute quotation. I mean, you cannot say I'm capable of detecting and then give a number. It is, of course, a function of the material. So the higher the crystallinity, the higher will be your level of detection. Not only, also it depends on your resolution in the case of crystal forms, and also whether you are detecting in a drug substance or not, because or in a drug product, because you have the interference with a lot of other elements, and then if you have a signal underneath another one, then of course your level of detection is compromised. So it can go down by 1 or 2 orders of magnitude, of course. And this is why we are also interested in exploring other approaches, and that's why we are collaborating with Rocco. This is a completely different approach of quantification of amorphous, giving us the possibility of discriminating between different amorphous. That's the challenge, because amorphous quantification is accessible as a whole. So by spiking, or there are classical methodology to do that, but then you do not distinguish which is which is a limitation. Yes, is certainly a big challenge.
And eventually, if I can continue on this, I mean, if you take back the example where basically we were getting this needle-like particle, I mean, how do we understand that out of an amorphous form, I mean, we reach through crystallization into a metastable form, while the material was plenty of seeds of the target form, so the most stable one. So it must have something to do with the molecular aptitude of the compound in this specific amorphous state in order to recrystallize into the alpha form in opposition as just, you know, taking benefit of all the seeds that were nearby just to enable crystal growth. So there is certainly some very interesting needs in order to further understand what are those implications.
And thanks a lot again. Okay, so it's I think it's almost time to close the connection. It has been a very nice cafe. It's a very special one. And so let me thank again the speaker. Let me thank everybody who attended this cafe, and I enjoyed very much. And I wish you a very pleasant time and very great collaboration for the future.
Thanks a lot, Cinzia. Thanks, everyone.
Good. Thank you, everybody. Thanks, Cynthia. Yeah.
Bye.
Bye-bye.
Bye.
Bye.